Everyone, thanks for coming in today. We are excited to welcome Sydney Smith. She's a postdoctoral associate at Yale in the Brain and Cognition Lab, and she earned her PhD in cognitive neuroscience at UC San Diego, where her work focused on computational approaches to understanding dynamic attention and depression, bridging clinical and cognitive neuroscience. And today she's going to be talking to us about her forward modeling, the neural mechanisms of human temporal expectations. So please join me in welcoming Sydney.
Thank you. Thank you so much for having me. It's really great to be here. And thank you for Kelly for thinking of me to invite. Today, I'm going to be talking a little bit about some work that I did in my PhD at UC San Diego, where I worked with Dr. Bradley Wojtek. And broadly, I'm interested in how attention fluctuates through time and how we can best study it in humans. And so I'll just get started. Okay. So our external environment is rich with regularities that we can use to anticipate and respond to the world around us. This can be something as intuitive as being able to synchronize your movements to the beat of music before hitting the dance floor, or anticipating the punchline of the joke that your PI just told, to something as consequential as making a left turn across traffic and trying to find the right timing to make that turn without getting T-boned. And so the research in this area generally falls into three main categories. Those are human behavior, neural mechanisms, and theoretical models. So we look at human behavior experiments. We usually see that people are using cognitive experiments to make manipulations of temporal expectations. And so these can include cued associations, which includes associating a particular cue with the likelihood of an event happening in time. Hazard rates, where merely the passage of time it increases the likelihood that an event will happen. Rhythms, where there are very predictable rhythmic structures where we can synchronize our expectations to. And sequences, where there are even more clustered, repetitive patterns within data. Most of human behavior is on these temporal expectation tests are measured in terms of reaction time and perceptual accuracy, where better behavior is associated with with faster reaction times and higher accuracy. You can also see some kinds of tapping synchronization, especially in those rhythmic attention tasks. So what I'll be focusing on mostly today are these rhythmic tasks. And the theoretical models that exist to explain human behavior on rhythmic attention tasks include these models that try to calculate these fluctuations of attention through time. And this includes some older models like dynamic attending, where attention is literally synchronizing and being modulated by these dynamic rhythmic expectations. They can be combined with Bayesian models, where our previous experience with these rhythmic stimuli improves our posterior estimations of when they'll happen, as well as some really creative dynamical systems modelings that could be based on like actual neural populations firing and inhibiting each other to generate these rhythmic expectations. Research on the neural mechanisms behind temporal expectation include research done in mice and rats mostly, where we see anticipatory spiking, like a ramping of neural spiking in anticipation of like some auditory tone happening. And in humans, we've studied this non-invasively, and we actually are able to observe the entrainment of slow frequency oscillations in EEG and MEG signals in humans and non-human primates. And so with this project, what I wanted to do was find a way to combine all of these things into one experiment. And so here I'm placing myself at the center of this three-way Venn diagram. And the way that I wanted to do this was this approach of actually thinking about the neural mechanisms, so starting with those ramping firing rates and hypothesizing how they might actually look in real awake behaving humans. And so the approach that I took was to simulate hypothetical field potentials that we could record in intracranial human subjects who are performing a temporal expectation task, and then compare them directly to the raw intracranial data. And so, again, I recorded data from 7 subjects with intracranial electrodes who are undergoing epilepsy monitoring for the condition. I had them perform a temporal expectation task that I designed specifically to manipulate temporal attention in rhythms. And then I used existing theoretical models of dynamic attention to generate simulations of neural activity of people performing this task. And so during this talk, I'm going to talk about the design of the temporal expectation task and the people who are performing it. I'm going to go into detail about the spiking models of temporal expectation, so what I think that neurons who are performing this operation are actually doing, and as how I went from those spiking models to simulate local field potentials that you can record with EEG or intracranial EEG, and then how I compared those simulations to real intracranial signals in humans to get a spatial mapping of the temporal expectation process and understands behavioral relevance. And so I'll start simple with this temporal expectation task. Okay, so this task is based on the idea that the more information that we have about temporal regularities and predictable structures in our environment, the better our timing judgments will be. So in this task, we present the subject with a sequence of auditory tones. These are just piano notes. And so they hear 3 to 5 isochronous tones. And the last tone in the sequence is always going to be jittered. So it's always going to be slightly early or slightly late. We then ask them to make a subjective judgment about the timing of that target tone, to classify it as early or late, and how confident they are. So if they are definitely sure that it's early, they would press 1. If they're definitely sure that it's late, they would press 4. And this task was designed by me and a collaborator, Keith Dolan, who's currently at the Institut Pasteur in Paris. So importantly, one of the manipulations that we did with this task was once we present them with the notes, we then can control the amount of information they have to make this timing judgment about the target tone by taking away notes from that sequence. So here, when they have 5 notes, they have the maximum amount of information. But when they only have 3, that final timing judgment is very difficult to make. And just to make this a little bit more intuitive, I'll show you an example trial of the task. So here you're about to hear 3 piano tones. Hopefully you can hear that out there, but we'll see. All right, and starting now. All right, can I get a response from the audience? Were you able to hear that?
No.
No, okay. Okay, well, they were 5 tones. The last tone was quite late, so it was like doot, 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 doot. And so hopefully you would be able to know that that last tone was definitely late. And I'll just skip that other one, but it's a 4-tone condition that's slightly early. Okay, well, the way that I analyzed their behavior on this task was using a sigmoid function or a psychometric function. And here on the x-axis is the jitter of that target tone, so how early or late it actually was. And on the y-axis is how likely the participant was to answer that the tone was late. And we can then bin their responses along the x-axis and get the likelihood overall that they would respond late. Perfect behavior on this task would just be a step function. So when that jitter is negative, they say it's always early. When the jitter is positive, they say it's always late. However, at those really low latencies, behavior it just gets really hard. And so their behavior isn't perfect. We can then fit a sigmoid function to their behavior. and get an overall estimate of their accuracy. So the slope of this sigmoid can then tell us how close they are to that perfect stub function. So steeper slopes, higher accuracy. And so this is actually real data from someone participating in this task in a 5-tone condition. So this is maximal information. But then as soon as we start taking away information, that slope starts getting shallower and shallower. And so when we do this in healthy participants, we see the expected trend of the steepest slopes in that 5-tone condition when they have the most information, and shallower slopes once you start taking those notes away. And you can see that reflected in a steepening sigmoid slope. When we did this task with 7 intracranial participants, we see similar behavior, slightly noisy, but that is actually quite expected from intracranial patients. They are undergoing brain surgery. They're in a pretty noisy environment, but we do see the same significant trend. And it's a very small sample size too. So now I want to talk a little bit about the people who are actually performing these tasks. These are patients who have intractable epilepsy undergoing intracranial monitoring for their seizure localization. And so they undergo neurosurgery and are implanted with hundreds of electrodes in and around their seizure locus. And so all of these electrodes are planted purely for clinical purposes, but then they wait in the hospital for up to about 2 weeks to have until they have enough seizures to where their neurologists, neurosurgeons, and epileptologists can correctly triangulate the part of the brain that their seizures are most likely coming from. But while they're waiting in the hospital, they're just sitting there waiting to have seizures. And so oftentimes they will consent to participate in research with us. And so I can go take my little piano tone experiment on a laptop and put it in front of them, and they can respond. And I can collect their intracranial brain data. So this led me to think about the actual neural processes that might be contributing to temporal expectation that allows us to make these very precise timing judgments in this auditory attention task. And so the first one is pretty obvious. It's just the incoming sensory input. Did they or did they not hear the tone? Another one might be top-down bias. So this kind of dynamic attention of when you expect the tone to happen based on the other two or the earlier tones. And then maybe there's some sort of sensory anticipation like procedure that's integrating these top-down expectations with those bottom-up sensory inputs in a way that allows us to make very precise timing judgments. And what I want to emphasize here is that all of these things are probably happening at once. And because they're happening simultaneously and with very similar rhythmicity, it's really, really difficult to be able to pull them apart. And so that is one of the goals of this research. So we have coverage in these 7 intracranial patients all around a lot of frontal, parietal, and temporal locations, which is perfect for an auditory task. And so now the goal is to think about these bottom-up, top-down, and integratory neural processes and where they might be happening in these intracranial patients. So now I wanted to think of the actual neural activity that might be generating these temporal expectation processes. And so again, we have top-down bias, sensory anticipation, and bottom-up sensory input. Let's start at the lowest level at the bottom-up sensory input. Here I used a neural spiking model based on the spiking response of the auditory nerve. And in the auditory nerve, we see an exponential increase in spike rate at the start of this tone in teal, followed by exponential decrease and then a plateau throughout the duration of the tone, and then it drops off. And this was based on literally the spiking response in the auditory nerve from a ferret, I believe. And so it's a super low level auditory input model. And so for someone performing this temporal expectation task, we might assume that their low-level neural responses might look something like this, where you have this increase in spiking quickly following the onset of the tone and then plateaus and then drops off. And you can tell there's a little bit of a delay between the onset of that teal bar, which is like the duration of the tone, um, and that neural spiking response, just because that it's about 25 milliseconds, and that's the amount of time it actually takes for the seven synapses in between the cochlea and this primary auditory cortex for that signal to get to where it's going. And then when I think about top-down bias, I think back to some of those models that I introduced at the very beginning in dynamic attention, where you have this modulation of expectation through time as kind of a gain control. And so I modeled this as a sequence of narrowing Gaussians, where the Gaussians get narrower and taller the more tones are proceeding. So the likelihood of a spike becomes more and more temporally precise the more tones pass, and more excitable the more information you have. And finally, for the sensory anticipation, I borrowed a model from a really beautiful paper in Nature Communications from the Jazzayeri lab at NYU. They created a dynamical systems model that inputs rhythmic sequences, and based on a mutually inhibitory relationship between an excitatory and inhibitory population, it reads out a ramping anticipatory signal from this Y component that looks something like this. And so you can see it as this kind of progressive ramping of activity leading up to the onset of each tone. So based on those models of spiking activity of someone performing this task, I was wondering, how do we go from the probability of a spike happening in time to an actual local field potential that resembles something that we could putatively record from a human performing this task? And so kind of go back to the basics. We have neural activity generates transmembrane currents and electric fields. And so these currents can be excitatory, where we have positive deflections in membrane potential. They can be inhibitory, where they're hyperpolarizing. There are also others that are like calcium spikes, different intrinsic resonance, and hyperpolarizations. But what I'm going to be focusing most on, and as the source of the local field potential and extracellular electric fields, are these excitatory and inhibitory currents. And so neural field potentials, like the type of thing that we can record with EEG or intracranial EEG, are thousands and millions, really, of these extracellular potentials happening over time. And when you have millions of these cells firing at once with varying degrees of temporal synchrony, they summate into something that we can record as a field potential, something like you could record with the intracranial electrodes or EEG. And so this is what I'm going to try to simulate from those neural spiking models that I talked about earlier. And so here I'm going to focus mostly on excitatory potentials, just because when we think of dynamic attention and anticipation, oftentimes this is an excitatory process. We're talking about neural gain and increasing the likelihood of a spike happening. So I then simulated local field potentials for each of those three models that I introduced earlier, and based on the parameters for each individual trial. And so in this hypothetical trial of 5 tones, I would think that this is probably what the probability of a spike would be for that top-down model. I could then convolve the spiking population with an excitatory postsynaptic potential kernel and get a putative voltage trace of 100 neurons participating in this top-down potential. When I sum all those up, for each one of these models, I can get an estimate of the local field potential for a population of 100 neurons participating in this neural process. So we have a population of bottom-up neurons. It might look something like this. Top-down bias neurons might be doing something like this. And sensory anticipation might look something like this. And so when I first started doing this and I was looking at these neural traces, I thought that they looked kind of ridiculous. But I wanted to see if they actually resembled any of the intracranial activity at all. And so how do I then compare those simulated signals to the real neural data that I was collecting from humans? So what I did was I just correlated them. I wanted to take that simulated signal and I correlated it with a Spearman correlation to that intracranial signal. And I was actually very shocked with how similar they could be. So here is one channel on the trial that highly resembled the simulated signal for the SAM model. And I would find similar channels that resembled the top down or the bottom up. And here on that bottom up signal, you can see that I included a strong negative correlation as well, because I think that the direction of the correlation could probably indicate like a similar temporal process, but it's just the sign flip. And this could represent a putative dipole in intracranial activity, which we often see, especially when we're recording, like, let's say, at the superficial edge of the cortex compared to a deeper layer of cortex. Okay. And so basically what I did was I simulated those three models for every single trial and correlated them to every single electrode in all 7 participants. And what I got was this kind of map of where my simulations matched the actual intracranial data. So for the bottom-up sensory input, I see a pattern that actually looks like what I might expect, where I have these highest correlation values kind of within Hassel's gyrus around here that is auditory processing centers. And here, the intensity of the color is representing that Spearman rho value, so the strength of the correlation. It's also the absolute value, so taking account of those dipoles again that I mentioned previously. And the size of the electrode here also represents the significance in a 1,000-fold permutation test. So if I randomly jitter the simulations, how significant were these values? And so when I looked at top-down bias, I see a pretty similar pattern where areas around the auditory cortex are showing some similarity, but we might even have some additional participation from frontal electrodes. And the sensory anticipation model had definitely some of the strongest and most distributed values compared to the other two. And so this is kind of a nice qualitative picture of what might be going on, but let's actually break it down by cortical region and compare some of these things. I'm also introducing an important comparison, and that's with a control signal. And so this is just the amplitude envelope of the actual sound that was being played. So it's something that has a very similar rhythmicity, but it's not necessarily a neural signal. And so on the x-axis, you can see I've included the different cortical regions. On the y is that absolute Spearman rho correlation value. I'm just going to be calling that similarity from now on. And there's a lot going on in this figure, and so I'll break it down by two of the most important regions for auditory temporal expectation, which are the prefrontal and the superior temporal and insular regions. It seems like in these two regions, the models that perform best are the sensory anticipation and top-down bias models. So the intracranial signals from humans performing this task actually resemble those simulated populations in these regions. So this is all cool, but when what does it actually mean? Like when an intracranial signal resembles one of these models, does it actually mean that the neural population there is performing that operation, is actually doing that? Is that cognitively relevant for someone performing the task? So to investigate this question, I selected a representative handful of electrodes. So basically one electrode per model per participant in the superior temporal cortex and insula, that region with kind of the highest values. And I performed a logistic regression to see if trial by trial, I could classify their accuracy on that trial from a combination of those similarity values for all those models and the control condition, as well as some properties of the stimulus itself for that trial. So from the very early results that I showed you with that sigmoid function with their behavior, we already know that the jitter of the trial is very important to determine their accuracy. And so I wanted to include those features as well to see if the neural data could actually give us any additional information about their accuracy on the trial. And so when I run this regression, I got some output, and here I've plotted the beta coefficients on the y-axis and each one of uh, the um, the effects on the x-axis. And so here you can see, unsurprisingly, that the relative jitter and the number of tones were very important for their classification accuracy. However, we also see that that sensory anticipation model, that kind of ramping dynamical systems model, also significantly predicted their behavior. So where it's more in trials where the neural activity was more similar to the sensory anticipation model, they were more likely to get the answer correct. All right. And so with that, this is a task that I designed to manipulate temporal expectation. Saw a effective manipulation of their classification accuracy and subjective temporal expectation tasks. I then simulated neural field potentials and directly compared them to intracranial data from subjects performing this task. I found that the simulated data resembled the actual intracranial data in regions associated with auditory temporal expectations, and those simulations correctly predicted their performance on the subjective attention task. Okay, and thank you to my advisor, my former advisor, Brad Wojtek, and all my lab members and everyone at UCSD, including the neurosurgeon, epileptologist, all the EMU nurses, and of course the patients, as well as some really wonderful collaborators who helped me with the data.